I'm Dave Baker. I'm head of sequencing at the Quadram Institute. I've been on the Norwich Research Park for nearly 30 years. Back in early 2020, obviously the COVID pandemic started, and the Quadram was approached to be one of the academic partners of the COG-UK Consortium. So basically that was the government invested, I think at the time, about £20 million into sequencing or looking for the variants and sequencing SARS-CoV-2. A lot of the labs at the time in the early 2020 were sequencing very low throughput, so high labour, low throughput. But I had this idea basically, so I thought straight away, why can't I pivot the methods that I've been developing for high throughput bacteria sequencing over to the COVID sequencing. So within a week or two, we were sequencing COVID samples that were coming into the building from the local hospital. The method I'd developed, which was Corona Hit, 10 times or tenfold cheaper than the alternative methods that were out there at the time. And in the end, over the sort of 2-year pandemic, we sequenced over 87,000 samples. It was said during the early stages of the pandemic, if Quadram were a country, we would be like the 5th country in the world in terms of the number of uploads of sequences to the COVID database. So it was quite impressive what we were doing in a small lab in the middle of Norfolk. During the COVID pandemic as well, we were part of the REACT study, which was real-time surveillance of the variants across the UK. In fact, we were the only sequencing hub in the UK to be sequencing REACT samples. And this had a massive impact on policy making within the government, which was quite important. So the Quadram Institute has become a national capability. In the future, if there was a similar situation with a pathogen or a virus, the method we have is sort of agnostic to the source. As long as we can generate double-stranded DNA, we can transfer this method over.